Hello friends, now I think it's a time for you people or we people to go out in the nature virtually. So we can have a typical study of flower. Okay, we can enjoy that. Just to understand what is flower, we need to understand what are the accessory words, which are the essential words, and how the flower is constructed. Okay, so just to understand basic concepts of any flower, we have to have an example for this. Okay, so this particular study we are going to finish with this particular species, that is hibiscus jaswand as a food, so which is very common. It's a garden plant, and each one of you have seen this plant. Are you right? So I think let us begin with the technical part of this. uh, this particular flower or inflorescence, you can say it's a flower with some reproductive structure and a bracts, and I think I have labeled the things. Now let us start with actual part only. So this is calyx, which is usually green in color. Then corolla means petals. Okay, so this is petal part. So these are accessory words. Let us think about the essential words. What are essential words? Essential words are those which take part in reproduction actively, means male part and female part. This particular flower can be called as a bisexual flower because both the sexes are present in a single flower. Now, let us think about the male reproductive system or male reproductive structure. So this is stamen with anther and filament. So this particular part is called as stamen. Similarly, female reproductive part, which is carpel. Female reproductive part, carpel, which is made up of stigma, style and ovary. Let me tell you in the beginning itself. Ovary, when fertilization occurs, there are certain changes in the ovary. Ovary becomes fruit. What about seeds? Seeds are made up of, they are produced from the ovules. Let us think about the typical lady finger. Have you seen lady finger? Do you like it? I like it very much. So always remember, when you take a section, then you will find 5 locules present vertically in a fruit. It's a fruit. It's a capsule type of fruit. Okay. So this vertical chambers, locules, they contain number of seeds. So practically we are going to learn the things, but let me tell you in the beginning itself, each locule contains some seeds. So arrangements are like this. So these are nothing but the seeds in matured fruit, that is the fertilized fruit or ready fruit, we can say. In the beginning, when we consider a simple flower, when it is not showing any fertilization stage, so before maturity, there is a continuous process of development, and in this process, ovary contains several locules. In this particular case, in case of hibiscus, that is jaswand, so there are 5 locules, pentalocular ovary. The terminology is pentalocular ovary. So this ovary has 5 locules, and locule contains ovules. So ovules are the particular site, okay, where pollen grain allow pollen tube to enter and further fertilization process occurs. I think most of you have studied this particular part in your standard 9 or standard 10, right? So let us revise things. Stigma, style, ovary. Stigma are the receptive plates, we can say, which are sticky by nature, where pollen grains which are liberated from the stamen, that is specifically the anther lobe, we can say, anther, and this is dehiscence of anther, the pollen grains are liberated and with the help of insect. So there should be some pollinator. So with the help of insect, the pollen grains get shifted to another flower or stigma of another flower. So then they start formation of pollen tube. Ultimately, pollen tube travels through the styler canal and it reaches to the ovule where egg cell is available, and male gametes further they fuse with the egg cell and they form zygote, then it further develops into embryo, and this is how the seed setting takes place. So these are the certain events which are there in any normal life cycle of angiosperm. Okay, I think we can have better pictures about this, so we can start with a little pause. So look at this picture, a typical plant, entire plant with a flower. Let us think about the flower only. Flower, androecium, that is male reproductive system, gynoecium, female reproductive part, and the leaves and the bud and the branches, stem, roots. So this is a typical representation of any flowering plant. Let us have a look at a typical bud condition. So this is a typical bud. Don't worry about apicalyx and pedicel. Simply think about how this particular bud is originating. And this is bract, a supportive leaf structure which is present along with the bud. Okay, so this is a typical bud structure. Let us have a look at androecium. We are talking about essential floral buds which take part actively in a reproduction process. So this is androecium, and this part you can consider gynoecium, entire part. So androecium, anthers, then anthers are with anther lobes and filament. Similarly, you can look at stigma and a style. I would like to share one thing, that we are considering this particular picture just as a beginning part of this chapter. Okay, after this particular explanation we will be switching to the demo table, because we are going to dissect the hibiscus flower, and definitely we are going to look at how anthers are arranged, how stigma, style and ovary appears there in a real flower. Because you people at your home can definitely dissect a flower. You can take a picture and you can share it with us. For as a sample, we'll share one picture of a dissected flower, and it is shared by one of your seniors. Okay, so let us continue with this. So this is androecium, gynoecium, typical parts, corolla, that is petals, and these are the sepals, means calyx, and apicalyx, ovary, everything you can see here. Let us continue with first, I think, so this part is anther part we are considering. So after dehiscence of anther, pollen grains they come out. So pollen grains of this particular hibiscus species are spiny. So these are the spiny pollen grains. Don't worry about the meiosis process and how the pollen grains are produced. We are going to learn that. But basically, think about how pollen grain appears. So it appears spiny in nature. So the outer covering of pollen grain is called as exine, and the inner coat which is present, that is called as intine. So just for a passing reference, I am mentioning these names. So this is spiny exine and this is a typical pollen grain of hibiscus. Let us have another picture for female reproductive part. It is gynoecium of hibiscus. So this is stigma. Then stigma, they form a long stylar canal through which the pollen tube passes, and this is a typical ovary region. Ovary, it contains ovules once again. So in this typical case of hibiscus, the ovary is with 5 locules, 5 chambers. In simple words, pentalocular ovary or ovary with 5 chambers, you can say. So this is thalamus. So ovary is placed on a thalamus. It's a cup-like structure. Once again, stigma, style and ovary. Let us have a section for TS of hibiscus ovary. Can you see the structure? It's a beautiful structure. I think you may get confused. Just to avoid your confusion, let me help you with parallel diagrams. So this is ovary and we are taking a section and this section is appearing here. Are you getting the point? So this is a locule, means chamber, then ovules, then central axis or a central axis which bears the ovule, which provides a firm support to the ovule. Then wall of the ovary, the outer wall of the ovary. So this is the wall. So this is TS, transverse section. You are watching the things vertically, and this entire structure is actual ovary. Okay. So I think we can have a pause here and we can switch to the actual demonstration table. So we have learnt about various parts of a typical hibiscus plant. So let us have a sample now. This is entire twig, okay, where you can find a fully grown hibiscus flower. Hibiscus flower attached to the mother axis, we can say, or a twig, we can say, with the help of pedicel. We have removed. We'll keep this twig aside. Let us have a close look at this particular structure. So this entire flower, you can easily identify this is bract, okay. These are apicalyx, these are the sepals, these are the petals. In other words, you can say this is calyx, this is corolla, or these are sepals, these are petals, okay. And this particular part, I'll show it here. So this is more suitable for each one of you to observe. This particular structure, a knob-like structure at end, 1, 2, 3, 4, 5. So these are the stigmas, okay. Then these small lollipop-like structures which are protruding out from a single tube-like structure. These are nothing but the male part, that is anther lobes along with the filaments, okay. So this is male reproductive system and female reproductive system. Both the things are together. So let us now have a look at this particular picture. So this particular picture we have learnt root system, stem, then the branches, then the leaves, and ultimately a flowering branch. So it appears like this. So this is a flowering branch where buds are produced, and then there is a typical formation of blooming of flower along with formation of male reproductive part, female reproductive part, and the accessory parts. Yes, here you can observe the bud. Okay, after this picture we can have a typical picture for general flower morphology. We are not going to learn each and everything in detail now, but this is your foundation course, and being in a foundation course and learning or revising the things in a better manner, you should recall the various terminologies. So this is a typical section. This is vertical section, VS. So it passes through a typical flower. You cannot expect a symmetry or you cannot expect a typical symmetry here. Okay, so this particular structure may confuse you, but if you look at this structure, you can easily find this is pedicel, a stalk-like structure. We are holding this. Then this is thalamus. Then this is sepal, that is calyx, and this is petal, yes, that is corolla. Then calyx, corolla, androecium, that is anther, filament means a stamen, the male reproductive part, and carpel, that is pistil. So it is made up of stigma, style, and ovary. So this particular stigma, where the pollen grains are liberated and ultimately they form a pollen tube, and pollen tube passes through a stylar canal and it reaches to the ovule. Now try to recall a typical diagram. I think it is not with me, but in later session we can have a look, because in your books many times it is shown that a typical stylar canal is present, the stigma disc is present, and a stylar canal through which the pollen tube passes, and a typical anatropous type of ovule is present, inverted type of ovule is present, and the pollen tube reaches to the egg cell and it forms, it shows a fertilization, and after fertilization it shows formation of zygote, embryo, and further ovary develops into a fruit, and fertilized ovule develops into a seed. So this particular structure which you can see there in your board books is a simple ovule, a single locular ovule. It is not like that. So as we have seen, this particular structure of stigma, that is pentalocular ovary, so it's a common thing. So many locules are present many locules are present in a single ovary, and each locule can be filled with single ovule or double ovule, and depends how variety develops itself in a nature. Okay, so this is a typical picture for diagrammatic representation of accessory words as well as essential words. Okay, now we'll consider this particular picture later. So after this, let us have a look at typical photograph. We have seen this. This is bud, and this in young condition, this part, green part, is called as calyx. This is calyx, and these are the corolla. What is corolla? Corolla means petals. So petals are always colorful, just to attract the insects for pollination, because this particular structure, reproductive structure, it undergoes cross pollination. So there are a number of anther lobes are present, and after dehiscence, a pollen shower, beautiful yellow colored or golden colored pollen shower occurs, and this particular yellow dust is getting attached to the body surface of any insect, which shows hairy nature of a skin, or you can say hairy nature of a body, and thereby the things are getting transferred to the another flower, and ultimately the pollination occurs and further fertilization process occurs. So these are the petals which attracts any pollinator, especially insects. Okay, so this is a typical bud condition with a bract. We have seen this is elongated portion, because this particular structure is well developed. Okay, now we can have another picture for anther. We are revising these things once again. So this is reniform anther. Reniform means renal system. Renal portal vein means anything which is related to kidney, that is renal. So this is reniform, which resembles its structure with kidney structure or externally. Okay, so this is filament. So reniform anther, and this is a typical staminal tube which is formed by a number of anthers, and these are the anthers which are attached to the staminal tube with the filament, and this is how the structure is overall present. This is stigma once again, sigma style and ovary. A typical structure of anther, you are well aware about this particular arrangement. So this is filament, this is these are anther lobes, and this is connective. So when you cut open this particular anther lobe shows presence of either 2 chambers or 4 chambers. So it is in the multiple of 2. Okay, usually you should remember this. Once again, these are the pollen grains, spiny exine. Okay, and lastly, let us have a look at or I can show it like this only. So this is typically stigma, style and ovary, and yes, we are going to dissect this flower in this particular fashion. Okay, are you ready? So let us begin with actual dissection. Now, so this is the most interesting work which I always enjoy, to dissect a flower in a very nice way. Okay. So here I am trying to separate. Here I am trying to separate a calyx let us see whether I am successful or not. Yes. So this is a crown-like structure. This is a crown-like structure. So this is nothing but the calyx, or you can say sepals, calyx or sepals. Okay. Now these are the petals. Petals, always remember, they are arranged in a twisted fashion. There are so many types of estivations. So this arrangement of petals and sepals on a thalamus is called as estivation. Anyhow, you can pronounce it. So this is a typical twisted type of estivation, and estivation, always remember, is better observable in bud condition. So I'll remove the typical structure, and hopefully I should not cut the ovary, and my cut should not be deep, so that easily I can remove the hopefully I have not cut the even if I tear off the petal, it is okay, because we should remove the things. We can have a close-up for this. how the things are getting removed. Yeah, look at a typical Venetian type. And always remember, when we say a flower is a modified shoot, means once upon a time this particular structure was acting as a leaf. It was green in color, and just to have a sexual reproduction, just to have crossing over, just to have multiple varieties of a single flower, the things are getting modified. So this is how the petals are also showing the traces of leaves. See, the Venetians are there, beautiful Venetian, you can easily observe. And this is reticular type of Venetian. Oh, by mistake I have dissected. Can you see this? Is it observable? For our better understanding, what I'll do, I'll simply remove the petals. I'm removing a petal. Okay, so this is how the petals are separated. I'm keeping them away from this particular structure, and let me tell you, once I remove it successfully, you can easily observe yes, now this is the entire structure. Yes, now we can have a look at this. Fantastic thing it is. So this is the most beautiful creation of nature. So this particular crown like structure which is present at tip is nothing but the male reproductive system and female reproductive system. Once again, so these are the stigma, this is styler canal. So I'll show you the canal, don't worry. And here will be the ovary. Now let us trace out a typical styler canal. Kaustubh sir, you can have a close look on this, because I think hopefully I can cut down the things, so this will help us a lot to get the entire styler canal. So the thing which I am cutting now is nothing but the staminal tube. So it's a tube which is formed by many stamens. So easily you can observe many stamens are here. And wish me luck. Okay, fantastic it is. Now I can say the things with more confidence that we have successfully taken out male reproductive part separate from the female reproductive part. So let us finish with this particular structure. So we can have a close look, and you can students, you can focus on the needle point. So these are the anthers. So these are the anthers which are attached to the original staminal tube with the filament. There are so many tubes and filaments are present, you can easily observe. So this is a fantastic thing. in the shadow, even in the shadow you can observe the things. Okay, so this is male reproductive system, and this is a female reproductive part, stigma. This is a styler canal, so big it is, and it is getting connected to the ovary. Stigma, style, ovary. Style forms a canal-like structure, hence it is called as styler canal. So better this can be observed on a black paper. So purposefully we are arranging the things in a better manner. Yes, so it's a beautiful thing which you can observe. This is styler canal, and great. Kaustubh sir, can you focus the things more if possible, because I know there are certain limitations of camera, but no issues. Still the things are quite observable. So these pollen dust are nothing but the pollen grains. So this yellow dust is nothing but the pollen grains. I'm not able to separate them, but still you can observe those yellow structures. They are dehisced one, these are open one, and the things are getting deposited on the stigma disc. So you can observe compared to the other structures, the stigma disc is more showing the presence of pollen grains, because they secrete sucrose solution, and which is sticky by nature, and this particular solution helps pollen grain to form a pollen tube, and pollen tube, it has male gametophyte, and we are going to learn that thing also. Okay, so this is the overall structure. Okay, then we can have a close look at ovary. So this structure is ovary. Okay, this is ovary, and now we can have a section for this. Okay, we can have a small pause just to arrange the things. So this is styler canal, and we need to remove it just to have a section. Okay, so this is a sharp blade with which we can take a section, and I'm sorry, I'm removing the things. Yes, I'm keeping the things aside. Let us have a section. Oh, look at the locules. I'll hold the things, Kaustubh sir, and if possible, please try to focus. Yes, you can observe the locules. So these are the locules, means chambers, which contain ovules. What I will do, I'll just press these locules. Don't worry, I can show you the section of a typical ovary by dissecting another flower. So we have a glass slide with us. So purposely I will put it on a black paper so that it will help you to observe the ovules. There are so many ovules. So don't worry that if your book is showing a typical single ovule for diagrammatic representation, so definitely the book setter, book writer, they have certain limitations, and definitely they cannot show all the things in a 3D manner. So let us have the things on a slide now. I'll separate out the things. So these are the ovules. I'm not separating each one of them, because this particular Malvaceae family, this Hibiscus rosa sinensis, means jaswant plant, belongs to family Malvaceae, and these are the ovules. Look at these structures, very beautiful structures. So this particular one seed like thing is nothing but the ovule. It is not a seed. When ovule get fertilized, it forms a seed, and whatever the ovary was present initially here on the thalamus, so this ovary will transform itself into a fruit, developed fruit. So these are the ovules. Have a close look, enjoy this, and try to understand how the things are there. We can have a typical section of ovary, and yes, definitely we can have a small pause to arrange the things. So let us have a section for typical locules. Okay, so I'm using a blade, and I have removed the other floral verse of another flower, another hibiscus flower, and now I'm taking a section. Look at the things carefully, and yes, at home you can try out with this sectioning, but be careful, you should not cut your fingers with a sharp blade. After all, you're a person who is going to be there in a medical college after 2 years, so you should be very careful with yourself. Then and then you will be able to take care of other people. Yes, beautiful sections we are taking, and we are going to use a small drop of water. Just to keep the sections moist. Okay, so this is the best way to keep the section fresh. I'm sorry. Okay, let's try out with some more sections. So taking section is a matter of practice only, so you need not to worry much. So let us have a close look under microscope for this particular section. So let us have a close look at the locules and how the ovules are arranged in the locule. So the thing which you are observing now is nothing but a section of an ovary. Okay, so it's a section of ovary, and you are observing a section through kaleidoscope. So this particular microscope like thing or magnifying instrument will be shown to you after this particular section. Okay, so let us have a close look. Yes, you can observe the locules. You can count the locules, you can count the ovules, the small seed-like structures, which you can observe are nothing but the these are nothing but the ovules, and ultimately when pollen tube will reach to the locule or near to the ovule will get fertilized. Okay, so this is a typical section. So this was a slide and this was a typical slidoscope, and what I did, I simply put this slide into this and I leave it. So this was a typical structure, and through which we were observing a typical section of section of ovary where we could observe various ovules. Okay. Now we are going to continue with the fertilized fruit, fertilized ovule, or developed fruit, or ripened ovary, anything you can call it. So let us have a lady finger. Just to expedite the things, I have already dissected one thing. Let me tell you, I'm going to cut this particular lady finger. Let us have a section first. So these are the locules. 1, 2, 3, 4, 5. These are the locules. This is central axis or placenta where the seeds are attached. Now understand the things carefully. So this was a typical ovule. Now it has become a seed, because this is nothing but a typical grown or ripened ovary. So this is a fruit. Fruit contains seed. Now originally this was ovule. So let us dissect the things once again for your better understanding. So I'll have straight one. So let us have a cut. Yes, we can have a close look at overall arrangement of fruit. So this ladyfinger, okra or bhindi, it belongs to family Malvaceae. So you can easily observe a stickiness while cutting this fruit, chopping this fruit. I hope this intact thing I'll be able to show you. Once again, you can perform all these practicals at home. So what are practicals? Practicals are nothing but the actual feeling of science. So just to have clear-cut idea about how the things are there in nature, we are performing practicals. Yes, in case of plants, we can easily procure samples and easily we can perform practicals. In case of animals, it is little impossible, and there are certain laws, so we have to follow the laws. And now we can have a look at various seeds which are arranged in a specific fashion on this central axis, that is placenta. And if closely, if you observe the things, so are you able to focus on this, Kaustubh sir? So these brown structures, okay, so these brown structures are nothing but the unfertilized ovules. So these are the fertilized ovules, means now they have become an independent seed, and this particular structure is nothing but the typical unfertilized ovule. So just to show you, I'll use forceps. I will remove them. I can collect them and I can put them here on a white surface where you can observe the things. Now, are you able to recall the structure of typical hibiscus ovule? So yes, we can show you both the things. These are the hibiscus ovules which are not fertilized yet, okay, and these are the in a white plate, these are the ovules of a typical ladyfinger, okra or bhindi plant, bhindi flower, which are unfertilized. If you compare these unfertilized with fertilized one, then you will find these are present. They are not showing any development, and there is no chance of getting fertilized after formation of several seeds and after ripening of the ovary, that is formation of fruit. So this is how the things are there in nature. So I think we have done with this small demo, and we are going to learn about how things are there with flowers, how anthers mature, how stigma mature, how ovary is receptive to show further changes in the structure, and how it allows a pollen tube to enter up to the egg which is to be fertilized in an ovule. So typical ovule structure we are going to learn, and I think in your syllabus up to 10 standard, anatropous type of ovule, the ovule which was inverted and pollen tube used to travel like this. So this was a typical structure, okay, we have learned this. We are going to learn fertilization process and then development and how the seeds are formed, we are going to learn.